coming to the next step is the elongation process. We know elongation. Elongation means here elongation means continuous addition of amino acids. Continuous addition of amino acids. So this is one of the fastest step in entire protein synthesis process. So elongation is the fastest step in entire protein synthesis process, right? During elongation process, codon recognition takes place. Codon recognition takes place. Codons are recognized by anticodons. Anticodons are present within tRNA structure. Then tRNA having specific amino acid. The specific amino acid based on anticodon. Anticodon recognizes the codon, right? So during elongation process, some steps are there. Again, some steps are there. The very first step is the codon recognition. Here codon recognition is nothing but this is one codon. This codon recognized by this anticodon. Again, this is the this is the P site, and this is the A site. See, always here this is the A site, this is the P site, right, and this is the E site. Always A site is supporting for the binding of coming tRNA structure. So here this is one of the A site. A site is supporting for the binding of coming tRNA. That is nothing but amino acid tRNA, right? So here one of the loaded tRNA bind at A site. Right? So here codon recognition. So what is codon recognition? Means codons are recognized by anticodons binding of loaded tRNA at A site. So the step is considered as codon recognition. Sir, which factors are supporting for the codon recognition? Two factors. One is temperature unstable factor. And next is the temperature stable factor. In prokaryotes, during elongation process, the very first step is codon recognition. The codon recognition done by two factors. One of the factor is temperature unstable. Other factor is the temperature stable. Right? These two factors are supporting for the codon recognition. Right? So this is one of the tRNA structure. The tRNA loaded with the amino acid. But suppose, see, this is the cyclic manner because this is the first codon, this is the second codon, this is the third codon. Continuously the codon recognition takes place. Continuously the codon recognition supported by the two factors, TU factor and the TS factor. TU indicates here temperature unstable, TS indicates here temperature stable, right? So this is the one of the tRNA loaded with the amino acid. So here TU means temperature unstable factor binded with the GTP. See that TU and GTP. See, these two factors are supporting for the binding of loaded tRNA at A site. This is the A site. This is the P site. This is the E site. So the loaded tRNA binding supported by one of the TU factor. TU factor utilizes the GTP. I already said every codon recognition requires energy. That energy here supplied by the GTP. The GTP binded with the TU factor. The TU factor binds to loaded tRNA. Then the loaded tRNA bind at the A site. bind at the A site, right? Once the binding is takes place during interaction, during codon and anticodon interaction, the GTP hydrolysis is takes place. The energy utilization is takes place, right? See, once the binding is takes place, the TU, okay, the TU dissociated, or the TU factor is released, okay, in the form of TU GDP, TU GDP, and inorganic phosphate PI. We know the GTP hydrolyzed into GDP plus PI. So here, same manner, the TU GDP, right? This is the one structure, and other one is the PI, inorganic phosphate. Again the TU factor loading is takes place. Loading is nothing but the TU here having low energy compound GDP, but always here energy is required. Always here energy is required. So again loading is takes place. The loading is carried out by the TS factor. TS is indicates here temperature stable factor. So temperature stable factor supporting for the binding of GTP, and releases the GDP. Simple. The TU initially binds to GDP. After binding of loaded tRNA at A site, the TU factor is released. Along with the TU factor, okay, here already GDP hydrolysis takes place, right? The TU factor having here GDP and inorganic phosphate is liberated, right? Again the TU factor loaded with the GDP molecule. The factor supporting here TS, the GTP loading supported by the TS factor. So TS factor supporting for the loading of GTP to TU factor. Then the TS also supporting for the removing of GDP. Internal exchange. See, one of the GDP is present here, one of the GTP is present here. The GDP is attached here. The GDP attached here, right? The GDP attached here. The GDP attached here, right? So exchanges takes place. Again, the TU factor is loaded with the GDP. Okay, this step takes place. In a cyclic manner. This is the first codon recognition, again the second codon recognition, like this. Okay, here all the codons are recognized. Right? This is the step, right? So this is the step, right? So here the final theme of this step is binding of loaded tRNA at A site. Binding of loaded tRNA at the A site, right? So in this manner, the codon recognition takes place. Codon recognition supported by two factors: temperature unstable factor and temperature stable factor. Temperature unstable initially having GTP. After the binding of loaded tRNA at A site, right? The GTP is converted into GDP plus PI. Again the TU factor loaded with the GTP supported by the TS factor. Again the TS, okay, binded with the GDP. The TS factor binded with the GDP. That's nothing but it removes the GDP, adds the GDP, right? So this is the codon recognition process. Right? This is the first step. And next step we'll discuss now. Yeah, the next step within elongation, the first step is the codon recognition. The second step is the peptide bond formation. All we know, peptide bond formation, especially done by the ribosomes. Within ribosomes, there is a P site is there. The P site contains peptide transferase enzyme. That enzyme supporting for the peptide bond formation. Especially in prokaryotic cells, there is a large subunit of the ribosome. The 50S subunit ribosome is there. That ribosome contains 23S rRNA. That rRNA also called ribozyme enzyme. That enzyme is supporting for the peptide bond formation. So how the peptide bond formation takes place, we'll discuss now. Very interesting fact is here, no requirement of energy for the peptide bond formation. No energy requirement for the peptide bond formation, right? See, after that codon recognition process, this is one of the formal methionine tRNA. This is the first codon, AUZ, right? And this is the first codon, right? AUZ, and this is the second codon. Here, second amino acid is recognized. Second amino acid is recognized. After the recognition process, so this is the site. This site is called as A site. This is the A site, and this is the P site, and this is the E site, right? We know about these sites also. We discussed so many times. See here, this is one of the P site. This is one of the A site. So between these two, there is an interaction takes place. Here one of the amino acid. This is formal methionine. This is formal methionine. The formal methionine here considers amino acid 1. This is amino acid 1, right? This is amino acid 2. This is amino acid 2. So between these two, peptide bond formation takes place. Between these two, peptide bond formation takes place. All we know the peptide bond means one of the amino acid contains there is an NH2. This is the H, and this is the side chain R, and this is the carboxyl group, right? This is the carboxyl group, right? See, this carboxyl group especially interact with the amino group of another amino acid. Amino group of another amino acid. See, this is the NH2, right? Again, this is the CH, and this is side chain R. This is the COO, right? So exactly the structure is like this. The structure is like this. See what I am telling here. Peptide bond formation takes place. Peptide bond means bond between carboxyl group and the amino group. Carboxyl group and the amino groups. This is NH. Exactly, this is the NH. So this one is called as peptide bond. This one is called as peptide bond. So here COOH is there and NH2 is there, right? Especially here one water molecule is removed. One water molecule is removed. What I am telling here, NH2, this is NH2, right? And there is an CH, this is a side chain R, this is the carboxyl group, initially COOH, right? And this is one of the NH2, right? So here NH2 indicates here NH, right? This is NH. Again, this is the next amino acid structure. This is the next amino acid structure, right? There is a carboxyl group is there and side chain is there. Carboxyl group is there and side chain is there. So what I am telling here, during peptide bond formation, one water molecule is removed. During peptide bond formation, one water molecule is removed, right? So here this is one of the peptide bond, C double bond O and H. That is the peptide bond, right? So the peptide bond, okay, carried out by the formation done by the peptide transferase enzyme. Peptide transferase enzyme. So that is about the second step within elongation process. That is about the second step within elongation process. Simple. After that, codon recognition process. Next step is the peptide bond. There is an amino acid 1 and amino acid 2. Between these two, okay, peptide bond formation takes place. Then amino acid 1 is shifted to the amino acid 1 is shifted to the amino acid 2. Amino acid 1 shifted to the amino acid 2 because between these two peptide bond formation takes place. This is empty tRNA. This is empty tRNA. Empty tRNA shifted to the E site. Empty tRNA shifted to the E site. Then, okay, the amino acid, the peptide bond contain tRNA shifted to the P site. This tRNA shifted to P site. This tRNA shifted to E site. And new tRNA binded, new loaded tRNA binded the A site. New loaded tRNA binded the A site. I will explain how the movement takes place, right? That is about the peptide bond formation. That is about the peptide bond formation, right? Here coming to the last step within elongation process, the step is the translocation. Translocation. It's nothing but movement of ribosomes on mRNA. Movement of ribosomes on mRNA. So here this is the one of the first complex structure, right? After the binding of subunits, initiation codon recognition. After that, next step is Next codon recognition. This is the first codon recognition. This is the second codon recognition. Okay, between these two. Okay, between these two. Right here, these two amino acids are there. Between these two amino acids, peptide bond formation takes place. Right. Next one is movement of ribosomes. Movement of ribosomes on mRNA is called as translocation. Is called as translocation. Always ribosomes are moving during protein synthesis process. Ribosomes are moving from 5' to 3' direction. Always ribosomes are moving during protein synthesis process 5' to 3' direction. This is also called translocation. Translocation of ribosomes on mRNA. Right? See how the ribosomes are moving. How the ribosomes are moving on mRNA structure. Here also one of the factor is there. The factor is called as elongation factor. Elongation factor. One of the elongation factor supporting for the movement of ribosomes on mRNA structure. Elongation factor supporting for the movement of ribosomes on mRNA structure. So here always the translocation of ribosomes on mRNA requires energy. Always requires energy, right? So the elongation factor binds with the GTP. The elongation factor binds with the GTP. That complex structure, that elongation factor, right, and GTP complex structure supporting for the movement of ribosomes on mRNA. So initially AUG presented T site. AUG codon presented the P site. After translocation, see this is the P site, this is the A site, yeah, this is the E site. E site, right? Initially, after translocation, see the AUG codon enter into the E site. Initially, AUG codon is present here. Ribosomes are moving in this direction. So here, whatever that P site contain codon is there, that codon enter into E site. The P site contain initiation codon enter into E site because ribosomes are moving. Then the A site contain the tRNA enter into P site. Then this is the new A site. This is the new means this codon is not recognized. This codon recognition is not completed. So coming amino acid tRNA bind at A site. So this process continuously takes place. Movement of ribosome and tRNA binding. Again peptide bond formation. Again movement of ribosome. Right? Again new tRNA binding. Right? Again peptide bond formation. So this process continuously takes place. So here elongation process, one of the step is the translocation. In this case energy is required. Means movement of ribosomes requires energy. And one of the factor also very important for the translocation process. One of the factor is also very important. The factor is called as elongation factor. Elongation factor always binds to GTP. Once the elongation factor or elongation complex binds to the ribosome, right? Here the GTP hydrolysis takes place. So by the utilization of that energy, movement of ribosome takes place. Once the movement of ribosome takes place, then here sites are changed. After that movement, see the elongation factor is there. The elongation factor releases the GDP plus PA. See hydrolysis of GTP forms the GDP plus PA. GTP hydrolyzed forms the GDP plus PA. Right? So here the step is translocation, means movement of ribosome requires energy, and one of the factor elongation factor, presence of these two, okay, movement of ribosome takes place. Right? So this is about the translocation, and one of the concept is here initial, means before the translocation there is an E site is there. This is the before the translocation E site is there, and P site is there, and next one is the A site is there, right? These are the initial sites. After translocation, the P site becomes E site. Because here ribosomes are moving on mRNA, the P site becomes E site. The A site becomes P site, and a new A site will be formed. And new A site will be formed. This is the new A site. This is the new A site. See, this is the this is the P site. This is the P site. So initially AUG present in P site. Initially AUG present in P site. After translocation, the AUG enter into E site. The AUG initially here at P site. After translocation, movement of ribosome, the AUG enter into the E site. Then the A site contains, here A site contains the codon enter into P site. And new A site will be formed here. New A site will be formed here. This is nothing but new codon. Okay. Again, this new codon recognized by the one of the tRNA. So already empty tRNA is there. The empty tRNA once enter into the exit site. Okay, that one is automatically moving out from the ribosome. Right? So that is about the translocation process. So by this mechanism, means we know about the very first step in elongation. Codon recognition. The next step is peptide bond formation. The third step is translocation. Okay, these are the steps which are seen in elongation. These are repeatedly happened. Finally, elongation takes place. Right? That is about the elongation.